Is that something people see?
I see your screen, but not your, there we go.
Do you see the slideshow?
I do,
You do?
yes.
Great. Alrighty. Well, without further ado, I guess I will begin talking about what I'm here to share with you all today. But just want to highlight a few things in my title slide. I am the current graduate assistant at the University of Idaho Stillinger Herbarium, and there's a little bit of contact information. And this is a wonderful picture from my first field season up here in the Selkirks, collecting up at the crest, and most of you might recognize Chimney Rock. It's one of the more charismatic features of the crest, and it's pretty wonderful to be up there, kind of in peak bloom, which is around late July, early August, up on the crest. And also, as Ken said, Ken Preston said, you can feel free to ask questions throughout the presentation. Anywho, I would really like to start today with an Indigenous land acknowledgement. I'm actually talking to you today from the Stillinger Herbarium, which is in Moscow, which is on Nimipu, Nez Perce land. But the area of the study is highlighted here, and it's actually an overlapping territory of the Kalispel and the Kootenai tribes of Idaho. You can kind of see that this whole blue area is the Kootenai tribe, which their Indigenous territory goes well into Canada. And then we also have some overlapping tribal lands with the Kalispel. And so both tribes were forcibly removed from their land in the mid 1800s, but lived here for hundreds, if not thousands of years. And so I'd like to acknowledge their presence in this area. But without further ado, the bulk of the talk will be about botany. Some of you have more experience with this than others, but I'd really like to start with this quote, which to me kind of sums up why botany is still important in 2020. But excited by the many new and rapidly growing fields of investigation associated with modern biology, we sometimes forget that is the actual plants, their ranges and other features and their status that we need to investigate first. Without the continuous acquisition of such knowledge, so fundamental for everything else, without the ability to recognize and distinguish the kinds of organisms, without the careful documentation of what we learn in biological collections, nothing else in systematic or evolutionary biology is even possible, which is by Peter Raven, a really amazing botanist from the Missouri Botanical Garden. But just to kind of contextualize my position here at the Stillinger Herbarium and that role within the project, and the importance of projects based in inventory. But I'd kind of like to just talk a little bit about the importance of natural history and collections in general. Our understanding of the natural world is entirely dependent on the physical record of specimens, and that's both flora and fauna. There's entomological collections, I'm butchering that, and from mammals to birds, to fungi to parasites. Here's some wonderful plant collections. And we can't actually have any type of, here we go with the poll. This is my first Zoom talk, by the way, so I'm hoping I'm doing it okay. But essentially, without any actual specimen, we actually don't have an understanding of the identification of such species, both in flora and fauna. And my talk today is going to be entirely about plants, and plant collections are housed entirely in herbaria, which is just a more specific base type of natural history collection. And so I put up a poll here, have you been to an herbaria before? And it looks kind of 50-50. Wonderful. Great, I'm gonna stop sharing this poll. So specifically, I'll be talking about the Stillinger Herbarium, which I think probably a few of you in this chat and this Zoom have been to before, but it's located on campus. Oh, here we go. Let's see who's been here before. Awesome. 7 people, 8, 9. Great. So I'll say not enough of y'all have been to the Herbarium down in Moscow before, but it's a really wonderful place, and it's where I'm currently located. You can actually see the compactor system behind me. This picture is actually from our open house that happened last year. We moved while I was here from one building on campus to the other, which was a pretty big effort. But the Stillinger Herbarium is the Idaho State Repository, which means that essentially, it's where you put plants. I'm just going to butcher this for pretty much essentially all studies within the state that happen both in and without in and around the state. And it's been around for a really long time, since nearly the establishment of the university in the late 1800s by Louis Henderson. And it was in one building on campus. It burnt down. And there's a lot of really unique history to this herbarium, and you should come on in and visit and learn a little bit more. But just to summarize, we're about a medium-sized herbaria with about 250,000 species, our collections, and most of those are vascular plant collections, and we have a few non-vascular collections. And we are still growing with projects like mine. Between 2,000 and 4,000 specimens are added a year. And a unique element of our herbaria are the tissue samples that we're growing here, and we have about a 4,500 tissue sample library. And on top of all of this, we also have a reference library that we are part of the university library system. And so we have a more focused botanical library. But I also just really like to highlight that this is where been the host of my project and of my time here in Moscow. I'm funded by the Stillinger Endowment, which funds my graduate fellowship. And on top of that, there's also this endowment, which funds not just my research, but others' research. There's other graduate students who use this fund and apply for these internal grants through the university to fund their research, which this project would not happen without. But we have undergraduate employees and local employees, as well as volunteers and oftentimes researchers. So it's a really great place. I will be here until the end of January, but Ben Legler is the collections manager and Dave Tank is the director. And we have our little email there if you're interested in getting a hold of us. So just in terms of the outline of the talk today, I kind of want to, I've already introduced the Stillinger, but I kind of want to talk about what floristics is, why I picked the Selkirks, how does one even execute this kind of study, what did I end up finding, why is it important, and then we'll end with acknowledgements and questions. Great, so what is a flora? I'm sure a bunch of you already know the answer to this, but for those of you who are unfamiliar, the flora that I'm referring to is essentially a list of plants, and all of those plants on that list have been verified and are attached to a vouchered specimen. And that list is attached to a defined geographic region, and that could be something that's really big. I have pictured here the flora of North America, but that could also be something small. It could be a county that has the defined geographic region by these political boundaries, but it could also be ecological. It could be a watershed. So there's some variability in what type of flora there is. There's also a few types of floras. There's the taxonomic key, like the flora of North America, that helps you identify plants. And then there's also the annotated checklist flora, which is what I executed in this study and what we'll be working on today, talking about today. Just as a small note, they're often targeting areas that are called floristic black holes, which are regions where there are insufficient collections to understand the flora of the area. And so I have a picture here of a charismatic black hole in northern Idaho that I actually targeted in this study. And you can see here, there's a density of collections essentially all around this mountain range, but not within it. And even though there are actual collections here, they're pretty concentrated on these areas around here. And so we have created a little bit of a whole. And this is a map that Ben Lugler created of all of the collections of the Pacific Northwest. And the green are collections made by that have been facilitated through the Stillinger Herbarium, and then the purple dots are ones, basically all other herbaria associated with the consortium, which I'll talk about later. But the typical elements of a checklist flora are the natural history, which some people include and some people don't. I did, and I think is invaluable to this research, and essentially your methods to your project, and then the actual list of plants. And there's many different ways to go about this. So why the Selkirks? The Selkirks, I'm hoping a few of you have been up here. I'm imagining a lot of you have. And they're kind of like nowhere else in the state of Idaho. This is a ecoregion map for the state of Idaho. And you can see that it actually is pretty ecologically distinct and defined as its own ecoregion. There's areas around it referred to as, with the Kootenai Valley or the Selkirk maritime areas. But the Selkirk Mountains itself are pretty distinct and also host some high Northern Rockies areas within their higher elevation parts of the range. The range does go well into Canada. This is just the southern extent, but this talk and my research is exclusively on the Idaho portion. But when you're targeting a floristic area, you're really looking for something that is attainable to fill in a period of time where you have the resources. So something that is, realistic to capture in the period of a master's. And then also, is pretty interesting to me as somebody who is just very curious around botanical history. So there's a really long history of botanical collecting, even though there was a lot of this area that hadn't been collected before. People started collecting here in the late 1800s, early 1900s, and some of those collections I was able to include in this study. But also that this area is really popular for recreation, and I'll talk a little bit about what that looks like. When areas are quite popular, they're often also potentially in danger of that kind of disturbance and development that could potentially harm some of those unique plant communities. And so when there is no baseline synthesis for the area, it also kind of begs to have a little extra look at it. The area is also unique, not just in its recreation and its history, but also in its assemblage of plant communities. Here we have a picture of the kind of general ecoregions of Eastern North America, and we can kind of see that it falls in this Rocky Mountain community generally, which is definitely true. And also it's has a few other unique features. It has a lot of this, this is Plisticum brownii, which is, quite way more prevalent on the coast, but finds it's a disjunct home up in the Upper Priest River Valley, up above Upper Priest Lake, and is one of many species that are coastally disjunct up here. And it's pretty interesting to work in an area that has such a strong coastal influence, even though you're quite inland. It's also quite north, being on the Canadian border, essentially, that we have a few boreal species dipping down. Some of you may recognize these cranberries. And then also there are areas of the mountain range that do have a Columbia Basin influence coming in from the south and west, that are a little bit more dry prairies and foothills. And then we have a little bit of this mixed Rocky Mountain influence that, is characteristic of the whole range, but there are a few more eastern species that dip over. So I guess I'd like to talk just about the study area. Here we have a map just of my defined study area. So it's 886 square miles. It ranges from the United States-Canadian border all the way down to Lake Pend Oreille, and is kind of cut off on its perimeter at the valley floor on all sides. It intersects a few different watersheds, it intersects a few different political boundaries. You know, here we have the Bonner and Boundary County lines. So there's a few different intersecting regional boundaries that are pretty interesting to engage with as somebody dealing with the logistics of this type of study. And then I also just, you know, I do spend a lot of time on the other end of the country, in the southern boundary, which is heavily militarized. And I think it's quite interesting spending time up at the Canadian border, where you just kind of have these warning signs, and there's not really much going on when you're in the wilderness, not wilderness designated, but when you're in the more wild spaces of the border. And I also just wanted to talk about the land ownership, and then also just about the range of elevation. On the perimeter, we're going again from the valley floor, and then as we rise into the middle, it's a little bit higher up. But the Selkirks are actually somewhat short of a mountain range. You know, they end at about 7,640 feet, which is an interesting feature to, you know, think is quite short. The subalpine area functions a little bit differently, being this north, but we do have true subalpine, but no true alpine up in the Selkirks. But then the land ownership you can see on this map is really interesting. All of this that is shaded is the Kinnikinnick National Forest, a part of the Idaho Panhandle Forest that the Forest Service manages. And then over here in the blue, we have the Priest Lake State Forest. And in the white, we have all state lands, and then a lot of private intersected. So there are quite a few different landowners and land managers that are intersecting within this area. In terms of logistics, fieldwork really begins with actually just getting the permission to do the fieldwork. We can identify a region, know that this needs to be collected in, but getting the permission and then getting the funding and the resources to execute such a study is really how we start. And so that really began with working with Jennifer Kostich Thompson with the Forest Service to get permits in order to execute this field work. And then it really began with trying to secure funding. I mentioned the Stillinger Expedition Fund and that endowment, and that's been crucial to this work. But on top of that, as Preston said, it's a pretty big range to collect by yourself. It's also very rugged. You know, I have to say a lot of people actually warned me against doing this project. They thought, especially as a woman, I would not be safe alone doing this. And so I was able to also, while I did spend a lot of time alone, I did have field assistants, which I was able to hire on through the grant and pay for their time, which was really awesome to also support budding botanists within this work, which was pretty crucial to this actually happening over these last couple of years. But a lot of logistics that I also interfaced with on top of these permits and funding was actually just getting the physical access to these areas. As Preston said, there's a lot that are just a lot of areas that are roadless, or a lot of areas that are in these areas called grizzly bear exposures that are managed by the Forest Service. And so they're just gated off roads that you are not able to access unless you have permits. And they don't even give internal Forest Service staff many chances to go beyond the gates. And then on top of this, there's a few really amazing research natural areas that have already been recognized for their unique species assemblages, such as Smith Creek in the Upper Pre-Scenic area, Three Ponds, and there were additional permits just to get to those areas. There, as I, kind of overwhelming you all with all of these amazing and ridiculous logistics to this this field work. One of the most pivotal was actually just being able to have a home base. Like I said, I'm down in Moscow, but over the past 2 summers, I was based in Sandpoint. And the first summer I was able to somehow slip by without a field station, and was able to make some friends within the community who had some space for me to put my field presses and my dryers. But this last year I was actually really fortunate enough to meet Kyle Nagy and be based at the Sandpoint Organic Agriculture Center. And you see that little yellow star. I was able to have a cabin for in order to host the project, which was a huge blessing to this project and the ability to engage in this field work. And some other logistics, you know, some people think about this, some other people don't, is things like weather and even just actually planning different types of approaches. The majority of my field work was conducted as day hikes into areas, but also I had, I think about 7 backpacking trips over the 2 years, between 2 and 3 days that I had to plan around weather and phenology, phenology being flowering time. So there's a few different logistics in order to make sure that that happens and I have the support and resources I need. But also it's a very unique range, you know, even though it doesn't have the bimodal summer weather plan, it does have some pretty intense summer weather, some of which gives way to really just a lot of mosquito, but other times to just full on snow and hailstorms in the summer. And this is us finishing collecting one summer during a hailstorm up on the crest, which was quite fun. But these are some just logistics I'd just like to mention that are vital to this work. And like Preston said, it's pretty much a unique type of field work, and that there are, you know, I think there was about a month where I wore a face net just for the mosquitoes this year, which the first year the mosquitoes weren't so bad, but this last year they were really they were quite potent. But also, yeah, there are a lot of areas without trails. You know, there's a lot of scrambling. Over here is just one area that we thought was an actual trail, but was just almost this vertical creek bed. This is up at Bog Creek. This is my field tech this last year, Moose, who many of you may know, lives in Sandpoint. And this is some of the areas of the sheer granite that can be a little bit scary and precarious at times. And that also just comes with field experiences that are unique. You know, we were up on the crest this one time and I fell down and the press exploded and I ended up just staying up late into the night. And one of my field techs captured this picture of me trying to put the press back together in my tent. But it, you know, amidst all of this, it's really quite worth it. So I don't want to freak anybody out. But I would like to talk a little bit about how to collect plants. You know, there are a few ethics that are important to mention. You know, there is this rule often associated with the permitting that, you know, within a stand of plants, you know, if you collect 1 for every 20 that you see. So there were times that I wasn't able to collect plants and I would document an elemental occurrence, but majority of the time I was able to collect. But there are 2 main types of plant collections that I would do. The first summer, I did a lot of general collections. That's referred to as the meander search method, where you essentially try and collect every single vascular plant species that you can, you know, and that's in flower or fruit, anything that you can identify and that you wouldn't harm the population. But then there's also certain types of collecting days where I would know I want to look for this plant. I think it's been up here and let's try and find it. And this is a picture of Moose looking for Botrychiums, these moonworts on a roadside, which is just more of a targeted taxon day. And then we also have the days where we're collecting everything and pressing lots and lots of plants. But also kind of would like to share how pivotal satellite imagery was in this project. You know, Preston asked me to share, how do you know where to collect? And this is not something that a lot of people talk about, but it is something that you kind of have to refine based on the region. You know, there's the obvious answers to this question. You know, we collect where it's never been collected before, which in this mountain range was basically everywhere that you can't get to, anywhere that had an accessible road had been collected, which is not very many. But then also the other obvious question is like, how do you get there? So the satellite imagery has been pivotal in figuring out, you know, what are areas that I can bushwhack through, what are areas that are realistic to climb, and then also what are targeted areas. realizing, oh, I think maybe the most diversity might be on the crest, where there's the most contiguous amount of granite, and then realizing I'm up there and it's all the same species. So there's a lot of chance, because you can't collect every single peak. We try and collect every plant specimen every plant species that's there in a representative vegetation. But there's some chance going into this. Sometimes you find stuff and sometimes you don't. It's a little bit of a balance act. But I would just like to summarize some highlights from each year. In 2019, I was able to spend 49 days collecting in the field, and that resulted in 2,090 specimens. Some exciting features of 2019 were the Idaho Botanical Foray. Most of you probably know Derek, but the Stillinger Herbarium, it was our turn to host the foray, and I was able to convince Dave and Ben to have it be up there in northern Idaho, so I could have it intersect. And it was really a great experience having people come from around the state, and some people collecting in the Purcells and the cabinets, and then having other people collect in the Selkirks with me. And this is a picture from the foray of all of the presses at the end of I think it was 3 days up there at that campsite. I was able to hire 2 field techs. This is Perry Lee Pipkin and Bo Romeo. This is up on the crest. I think it's just a wonderful and charismatic place within the Selkirks, and is definitely a highlight. And then some unique species we found that year, this club moss up at Lion's Head that Ben spotted within this heath field, and then this really amazing bladderwort I'll talk a little bit more about later. But you can also see, I got a little bit of a later start. I was still taking classes that semester, and so I wasn't able to do as much as I'd like. But I was still able to get a good amount of coverage that year. And in general, in 2019, I was doing a lot more general collecting. And then the next year, I was able to do a lot more targeted collections. I was able to start a lot earlier this year, and even amidst the interjection of this pandemic, we were able to still be really safe about field work, which is one of the privileges of doing botanical field work. I was able to hire Moose, who became one of my, few friends up in the area, and is a wonderful budding botanist. And Ben was able to join with me a few times, and I got way into moonworts this summer. And this is also one of my favorite plants, Gaultheria hispidula, and those fruits, they're just pretty striking how white they are. But I was able to spend a little bit more time up there this year, especially the biggest piece being having that field station, and able to round out the collections, which was really nice. And so in plant collecting, there's some people who are quite afraid of collecting plants. There's some people who have, maybe not the best ethics. And that kind of mirrors the fact that within how we collect plants, there's definitely a variability. There's some softer plants that you collect quite delicately, and then there's some where you can knock them down. I made these little videos of my mentors collecting plants. This has been collecting Goblin's moonwort. You can see it's quite a delicate plant. And this is after we've already assessed the population, and we can say definitively that we're not going to harm it. And then there's also things like aquatics, where you are floating these plants out in order to mount them properly, so you can see all of their parts, which Ben was a great teacher in helping me learn how to do that. And then there's also some, more classic styles. This is Dave, my other mentor, showing me how to get some cottonwood buds off the tree in order to make a cottonwood collection. So there's varying ways of collecting plants. Okay, so I also want to talk about something unique that I was able to do with this project was and how I was collecting the metadata attached to every collection. Most people who have collected plants do it with pen and paper. They collect their plant, they put it in a plastic baggie to press later at the car or at a field station, or they press it in the field with a field press. There's many ways to go about this, but they're taking their site data and their GPS and everything on pen and paper, which has been done for hundreds of years and is a foolproof way of collecting. However, with my limited timeline and my interest of trying to get the scale of collections I wanted, I was able to use a really kind of, you could say, modern way of collecting the metadata attached with every plant, which was through a mobile app that Ben Legler, one of my mentors here, created, and it's unpublished, but it is directly tied to our internal Stillinger Herbarium database. And from there, I can then edit those collection entries and then automatically publish them to our public consortium website, which is a pretty cool way of collecting. It enables you to go a little bit quicker around some of these things. But you can see over here that you're essentially making a site, and you can give this site the same amount of information you would give anything, but it goes a little bit quicker because we have presets for where we are, and it automatically gives each collection a number, and I can make the sites have anywhere. I think my sites, sometimes I had sites that only had one plant collection, but some sites could have we're up to like 60 plant collections in one site. So it kind of varies dependent on the phenology and what you're seeing and what type of collections you're doing, if it's targeted or general. But this is a little bit of a look into the interface. This is an individual collection entry that you can see. And then this is a little bit of the database, and I'll talk a little bit more about the consortium later on, but it's been quite cool to see my collections automatically go live and already have people emailing me and talking about these collections already being accessible. But for those of you who are not familiar with the collection process, I just wanted to go through it, how you take a plant from the field and then how it ends up in a specimen in the stacks behind me. So this is a fun pick. I chose this one just because I really like Phagopterus connectilis, this really cool fern. It grows in these really mesic, old growth Western red cedar areas, and then also all the way up to the crest, which is pretty cool. And this is up on the crest this last summer with moose. So essentially you begin your collecting with the actual physical collection. I put this one in a bag, and then once I got to my base camp, this was during a backpacking trip, here I am pressing the plant. And then a few days later, I was able to transfer my field press to a full press and put it in the dryer. This is a plant dryer, and it's essentially just two space heaters in a box that help the air flow through and comes out through the cardboard. And it's a pretty simple system that most people could probably figure out. But this part is really the quick piece. There's a pretty huge labor of love that goes into collecting specimens and bringing them from the field and then having them ultimately be something that's fileable into the collections. But from there, I'm able to bring them back to the herbarium themselves. I freeze the specimens so they don't bring in any pests or diseases that could potentially get into the rest of the collections. And in my case, because I am working with thousands of collections at a time, I sort them by their families. So I can key by group and also see a lot more patterns within the group's variability. And so I won't miss things like infrataxa, such as subspecies or varietals. And this is a little bit of a picture of my station that I'm actually currently sitting at, where I identify the plants. This is a microscope. This is the physical plant in the newspaper. And then I have my taxonomic key. And then I often have a few other keys pulled up on browsers from different databases, from the Flora of North America to the Jepson. And this is a spot that I sit at a lot and is often the big stall within plant specimens going into herbaria. A big piece of my work here has been not just identifying my specimens, but updating the specimen identifications within the Stillinger holdings. And so on top of using the resources like a treatment, I'm also using the physical specimens themselves. And so, I kind of put this here as like, choose your own adventure. You could sit down and identify plants, but you could also get caught in a taxonomic wormhole where you're interested in some updated concepts. You could also just not get an answer. You could be like, this specimen doesn't fit anything, or you could have a clean annotation, which, is what we aim for most of the time. From there, once the specimens are identified, I'm able to print and insert labels into the specimens themselves. We have a wonderful undergraduate staff who works on mounting the specimens. And then we accession them into our own herbaria, and that is marked with the accession number and the barcode. And then they're also imaged, which is a really great feature of modern herbaria practices. And then they're actually finally filed into collections. That's not just the end piece of them. When a plant is filed into collections, it's not a static holding. It's something that, I then reference, others reference for years to come. I've even annotated some of my specimens from the year before. So they're, while they're filed into the collections, it's not the all end for them. But the next piece I'd really like to talk about is the actual identifying. So this is a picture of my cabinet. I have 2 cabinets, but just what it looks like, all of the specimens organized into family. And this is most of the resources I'm using in order to identify these plants. The big piece being the actual reference specimens here. And it's why being at the herbarium is pretty vital. But one of the most important pieces was the updated Flora of the Pacific Northwest, which has been, an invaluable resource in order to understand a lot of the updated taxonomy. I learned botany with the Jepson and then with the last Flora of Pacific Northwest. So it's been pretty wonderful to then also be working with somebody who wrote a good amount of the keys. And I'm talking about Ben, and it's been really nice to learn a little bit more also about the ways in like the architecture in which a good key is written. But like I said, it's not always, it doesn't always end up in just every plant is cleanly identified. And so there are some issues, and I handle them case by case. I kind of want to encourage other folks struggling with plant identifications that if they are in a group that's messy, that there are a lot of publications, just looking up on Google Scholar, who is the most recent person working on this group and what are the most recent understandings of this group has been really helpful. And so pairing publications with treatments through time has been really helpful to me. But I also didn't do this alone. And I've had a lot of assistance through experts, especially on difficult groups. A lot of you probably know Barbara. She's been really helpful with potentias and roses, especially because the area I work in is the area of hybridization. Ben has been very helpful generally, but he is quite good with moonworts and with sedges. Mary Barkworth confirmed a really exciting elemus that's in addition to the area. Don also confirms moonwort. We had Mark Egger work on some Castanea, and then Carl Taylor worked on an Isodes, which was, it's been really interesting to work with experts in the, and to have their confirmations. And historical specimens as the other piece, I collected, over 4,000 plant specimens, but I also missed some species that were collected historically. This is actually a picture of an Isodes occidentalis collected in 1901 at Priest Lake. It's a really beautiful specimen. I just wanted to highlight the label data over here, but the other half of my project has been verifying historical collections of taxa that I didn't collect. And so there's been around 2,800 collections throughout the area that I'm studying. And my general estimation is that I have looked at about 1,000, and that's stuff I either verified or I annotated over the last 2 years. And so there's a large bulk of specimens that aren't mine that I'm working on. And so through these specimens, there's been just also, there's been a handful of patterns from blatant misidentifications, but also nomenclatural changes through time in the way we understand plants. And then also just kind of the patterns of the same groups that I struggle with, seeing botanists struggle with them, 50 years before. And right now I have 66 taxa that are represented by historical specimens that are being added to this checklist. And those are housed at the Stillinger Herbarium at the University of Washington, and then at the Marion Ombie Herbarium in Washington State. And all of this could not happen without the Consortium of Pacific Northwest Herbaria, which enabled me to not only identify the area that I'm choosing as in the Selkirks, but then in addition, identify all the collections and work through them. And I think it's going to be a big feature in people working on floras of this nature in the future in order to integrate the historical collections. But I'd kind of like to talk, move the general feature of what we're talking about more to like my findings now, especially as we're moving towards the end of the talk. That's kind of been all of the methods, which is definitely a mouthful. But a big piece of this project has also been in learning the natural history of the area. And the sections I listed here are the ones that will be included in the document. I don't have time to talk about them today, but I think they're pretty pivotal. A lot of people ask, why do you care about the settler history for the area, and how does that intersect with these subalpine environments? And I think it's, there's no way to separate the two in many ways. And this picture that somebody tagged, the flat earth up at Beehive Lakes, that intersects up into the crest up there, you have Twin Peaks, is a pretty charismatic picture for me to kind of explain why natural history matters in our intersection of botany. And I would like to briefly talk about the geologic history. This is a picture up at Bonner's Ferry, and you can kind of really see the large change within the landscape around us. But the Selkirk Mountains were formed through uplift. And so that was essentially this form, and this was millions and millions of years ago. And I'm not so good at geologic timescale yet, but this uplift basically encouraged the rock that was once on top of the Selkirks to move east, and is now the rock that makes up the Purcells and the Cabinet Mountains. And that through the uplift, it also formed the Kootenai Valley floor. And so that was one of the big major events in the geologic history. And that happened not just because of the magma, because of these faults that occur here. Many of you have probably heard of the term the Purcell Trench, which some people use ubiquitously to refer to the Kootenai Valley floor. This one event was a huge shape for this area, but then on top of that, there was glacial activity. I'm sure a lot of you have heard of the continental ice sheet, which came down and drifted over this area and formed a lot of depositional areas, which resulted in alluvial areas. And in addition to that, there was also alpine glaciation, which through erosion kind of carved out some of that charismatic granite crest that we see. And so this picture is really cool. It's from a 1956 survey of the area, but all that is orange, it was covered in the continental ice sheet. And you can see that kind of stops around Lake Pend Oreille. And then what is red is where the alpine glaciation occurred. And so those two types of glaciation, both the alpine and the continental ice sheet, resulted in the formation of two really interesting vegetation types, one of them being the granite Selkirk crest, and the other one being some really cool peatlands, which I'll talk about in a second. The climate of this area is also really unique. A lot of you have probably seen a lot of the maritime influence to the area coming in from the coast. This is actually a picture of Ball Lakes up up high up, kind of not too far from Roman Nose area, fully covered in fog in the middle of August one summer. And, you know, this is about mid-June, you know, there's just hiking through the snow. So the area is characterized by a northern rocky climate, which is the dry summer and cold, wet winters that kind of at high elevations can pervade into the higher elevation areas. And now I would like to kind of transition into the vegetation types. And I think we have a poll here, which would be a good moment to take a sip of water. Great. So while you guys are answering this poll around what vegetation class holds the most diversity in the study area, a part of my project on top of collecting these plants has been characterizing the mountain range and the vegetation within the area. And so I decided to characterize the vegetation within the area based on a model provided within the National Classification of Vegetation through the USGS. And so I've used 5 classes, which resulted in 17 types. And so this is an example. The class is wetland, but the specific type is deciduous riparian. And so I had these 5 classes, and I was curious what you all think holds the most diversity in the study area. And a few of you are still answering, but it seems like you all are picking up, you listen to the geology section and realizing that the wetland area where those peatlands are, where there's a lot of the products of that glacial activity, do host a lot of diverse species assemblages. So whoever said 3 is right. It is in the wetland aquatic zones. So I'm just going to briefly go through these vegetation types. We have forest, which a lot of people are very familiar with in this area. It's a highly forested mountain range, and this class ranges from lowland mountain forests to subalpine conifer forests, and is well known for having the largest contiguous stand of old growth western red cedar. And this is actually my advisor Dave standing in it up in the Upper Priest area. Then we have grasslands and shrublands. This class ranges from lowland montane grassland shrublands to fairly charismatic bare grass meadows that come to like mid-high elevations. And this class was one of the least documented classes for the region. So while it's not the most diverse, it was still important. And then we have our wetland aquatic riparian class, which has the greatest diversity, not just of vegetation types, but of species assemblages. And this ranges from lowland fens to lakes up to subalpine meadows. It also hosts these peatlands, also known as flow-through fens, and some of these areas are the only vegetation types in the whole state, and they exist all in the Selkirks. And a lot of people are probably familiar with hearing that, you know, the peatland habitat type hosts 10% of the state's rare flora, so they're pretty imperative that they continue to be conserved. There's also a few more subtypes that aren't even listed here, but just to give you an overview, and then also show you some unique subtypes like the floating mat that actually floats on these water features, which are quite fun to play with. Next, we have the open rock class, which ranges from mid to high rocky outcrops to the ubiquitous crest. We have some rhodiola and tigrifolia up here on the granite crest. We have fell fields, and then we even have talus rocky outcrops and slopes. And most of these areas are pretty characterized by these large openings that don't have much tree cover. And also don't have a lot of vegetation themselves. But they're also pretty resilient areas with some pretty intense weathering. So amidst all of that, there's some pretty unique species assemblages in these open rocky areas. And then lastly, the last class is the the disturbed class, disturbed being anthropogenic influence. And we have some roadsides that I've probably made look much more dreamy than they actually are. And then also, you know, there's an over century long history of logging in the area. This is my field tech from 2 summers ago, Perry Lee, and you can see this patchwork assemblage of all of the different logging. This is over on the state land side. And this was really prevalent throughout the study. There can be a lot of non-native species, but there can also still be native species. And then there's also a lot of development within the area. And a lot of you have probably been up to Schweitzer. There's a ski resort within the mountain range. No joke. I thought that was funny. I think there's a pole here, but yes. So I am asking you all, what type of plant is the person in this picture looking at on the roadside? This is, you know, more just for fun than anything. But we've got some split guesses. You know, this is not a test. I haven't told you anything about this. But I think we all get it. And it seems like most people, oof. Yeah, I think we're looking at a flower or a rare noxious grass, which are great guesses. But actually, there aren't any rare noxious grasses within the Selkirks. And this person's not also looking at a flower either. It is actually a type of fern that we're looking at, which I really just included that poll to let you know that even within all of these disturbed anthropogenically influenced landscapes, there is some unique diversity. And there's some pretty cool species that you can even find on roadsides within these areas. But I really love to actually just begin summarizing my results, which are preliminary for I'm still not finished with the study. I'm still working on it, but this is where I'm at right now. And so just there's a lot of information here, but some things I'd like to highlight are the actual taxa of this range. So while there's, you know, thousands and thousands of plant collections, I'm also collecting distribution data, and it, you know, I don't need to collect one plant once. I need to see what type of vegetation and features that plant can grow in, how resilient it is, if it's only, you know, found in one area. And so the actual numbers we're really looking at for the species in the Selkirks are 838 for that defined study area. And so of that 838, 772 of those taxa are ones collected by me, and 66 of those are historical. And so I just have some basic summaries of what I was able to find up there. One of the coolest things to note are the 5 largest families, which are pretty common. Most people would assume that the Asteraceae, Poaceae, Cyperaceae, and Rosaceae would be some of the more common and diverse families up there. But the Ericaceae is pretty cool. I think really points to that northern boreal locality for the study area. And then in terms of the genera, I think it's also really cool that how many sedges were found. This is a picture of Carex magellanica, which is another rare species, one of my favorite up there. There's a lot of sedges up there. It's pretty remarkable. And on top of that, within the flora, we have, I think actually it's more like somewhere between 95 and 99 not introduced species. And so the native taxa that we're looking at for the Selkirks is around 740, just a handful of noxious species, and then a lot of rare species. I was able to collect 4, potentially 5, waiting on some confirmation, plants that have never been collected in the state of Idaho before. And then I actually did my first actual summary of the county collections, and there was about 100 taxa that hadn't been collected in either county before. So that's still preliminary, but that's my general estimate right now, which is pretty amazing. And this is a very overwhelming slide. Please don't try and read this list, but it's really just here to demonstrate the extensive amount of rare species that exist in this study area. There's 54, 11 of which are S1, which is state imperiled, and 5 of those 11 only exist in the study area. And within here, there's many S2s that are also only listed and exist in the study area. And many of those are only known to one population. Some of you may be familiar with the sedge Trichoforum alpinum, or the heath shrub Andromeda polifolia. There's some bladderworts, this really interesting oxalis, a violet. There's a lot of really cool species that really only grow just there. And they're tied to a lot of these unique vegetation types, which are pretty unique for the region. There's also even some unique stories within the introduced taxa. This is a picture Perry Lee took of me walking through a field of noxious species, which is kind of funny. 2 of them being Hyricium aurantiacum and Hyricium glomerulatum, which were previously overlooked in the state of Idaho. Luckily, most of the introduced species are pretty concentrated to disturbed areas and haven't infiltrated too much into some of the less accessible areas. But that's a pretty high number of introduced species, but also contextually makes sense for how large the region is. And I think I have a poll here. I think I have a few polls because this is one of my favorite group of plants. Don't worry, we're almost done here. Yay. So this is just a total wildcard attempt at seeing just people's understanding of basic plant ID and guesses. Which one of these species is introduced? Guesses? I don't know if people can see the poll. While you all are doing this one poll, you'll see that there's 2 questions for this slide. Not only which one of these species is introduced, but then also which one of these species has never been collected in the state of Idaho, and you can have some guesses. And there's no such thing as a wrong answer. This is really just to see what people are thinking about these plants, if they've even seen them before. Great. Well, let's end this poll and see what people think. Well, y'all are right. One of the only introduced species is this Euphrasia nemorosa, which is a great job. Most of you probably know that plant as eyebright. And then which of these species has never been collected in the state of Idaho? Most of you all there's some mixed results, but actually, it is all of these plants have never been collected in the state of Idaho, except this one, this moonwort, potentially. We're still waiting to hear back from some correspondence. But this is a summary of the species that were not collected in the state of Idaho before the study. And were really great species to find. And I will be writing up in a Madrono article. And they were all kind of found in some pretty unique environments. This is a really cool club moss that Ben actually found up on the crest that kind of easily mixes in with the Cassiope and Phyllodoceae up high in those heath meadows. This is a really great Elamis hirsutus. This has this really amazing nodding spike or nodding panicle. And some really interesting ciliate hairs on the lemmas that grow in these kind of more mesic forests. This is up at Smith Creek. And then we have this really wonderful bladderwort that also was previously misidentified, which is pretty common, but a really wonderful bladderwort that occurs up in these peatland bogs. And this is one of our an invasive, or not invasive, an introduced species that grows pretty ubiquitously, but had never been collected in the state of Idaho, which just kind of gives you an understanding of how overlooked some species can get And this is a really great moonwort, Botricium hesperium, that was actually collected right up at the border near Bog Creek, near American Falls. And we're still waiting on confirmation if it's been collected in the state before. But right now, you know, a lot of these species, the general pattern is that they were essentially overlooked and they fill the hole within our gap of understanding of this region. There was also some plants I didn't find that historically were found, and they were up in these high microsites within the crest, one of them being Luetzkia pectinata, which is pictured here. And so you know, there were also, for every species I didn't find, there was a species I ended up finding that I didn't expect to find, one of them being Dryas hookeriana, which is a really cool higher elevation rosaceae species that was found right up in this tiny little pocket. This is up at Twin Peaks. This is Moose kind of overlooking one of the large scale reliefs of the Selkirk crest on over to the Priest River Valley, Priest Lake Valley. So some of our guesses is, you know, maybe these species were more widespread. Now they're restricted. Some of these species that I didn't find, you know, do they still exist? They haven't been relocated since the 90s. Some of these, the Luetzkia and the Luisia, haven't been seen since the 60s. Up there, you know, and these are species that really, the conditions have to be right. They have to be perfect. The snowpack and weather just has to be just right. The soil conditions, and, you know, a question that we have to ask is, you know, is there enough time within a 2-year study to capture and find a species like that? We don't know. And we also know that because these mountains peak out at 7,600 feet, you know, there isn't an area for them to climb with a threat like climate change. So they may not be occurring here for much longer, but we can't definitively say that either. There's also a class of plants I didn't find that were pretty concentrated to the Priest River experimental forest made by Daubenmeier, this really amazing botanist from who did a lot of work in the 40s doing vegetation typing of northern Idaho. And then also Peter Stickney with the US Forest Service. And most of these collections are up around the Priest River experimental forest, which, if you recall those early maps, had a lot of collections in the beginning. And then another person who really contributed to the historical collections for the area was Robert Bursick, and he has about 7 or 8 sedges that I didn't, that I overlooked amidst the 58. And without their work, you know, this project wouldn't happen, and it wouldn't be quite as thorough. And a lot of these species are ones where they're not everywhere. They're ones that I either overlooked or are potentially not there anymore. And this is an Atriplex patula, which is much more of a foothills lowland species that was found up at the experimental forest. And these ones that could have been brought in through the nursery or through logging trucks. And so they're not ubiquitous. But on top of that, I will be creating an excluded species list of some of these non-native historical species that were found on the edge of the study range or were historical specimens with a vague locality. If you're from the area, you know, if somebody says Pack River, you could be referring to, you know, about 15 different localities. And so there's some areas we can't definitively say are within the study area. And so I'm compiling those for an appendix. And lastly, some cool plants I didn't expect to find were some of these species that have range extensions. These are this really cool monkey flower, Diplacus clavicula, and this really wonderful lupin. These are both found in the southern extent of the range, some near Riley Creek and near Blue Lake over on the state side. And you can see in both these maps, there's this large cluster, and then there's a dot quite up north in the Selkirks, and those are my collections. And these are both found in these montane areas that were completely under-collected. You know, a lot of us wonder, you know, is this a relictual species? Is this a northern expansion post-glaciation? But it also is great data for other people to use in phylogeographic studies that could happen after this. But it also just points to the fact that a lot of these areas were just actually under-collected and we didn't have a full picture of the area. Some of these lists of really common species were never collected in both counties up in the northern Idaho panhandle. And so we really just needed to get up there and do some general collecting in order to fill the whole of the montane areas. And so I'm going to start with some of the kind of wrapping up. These are the collection sites. And so every dot on this map could refer to anywhere between 1 to, I think, sometimes 60 species. And so these are areas that are collected in the northern Idaho Panhandle before the study. You can see they're concentrated around Lake Pend Oreille, around Priest Lake. And this right here is the Priest River Experimental Forest. Over here, you have the Kootenai Valley. This is what my study was able to complete within the 2 years. And so this is every site that I collected at between 2019 and 2020. And then this is all of those sites together for the study area. So amidst all of this, I was able to cover a lot of ground, able to find out a lot more. A flora may still never be complete. A lot of people talk about a species name actually just being a hypothesis, which I'm sure a lot of amateur botanists hate to hear. But a flora is technically never complete, because species are entering and species are leaving, both with development, with climate change, with introduced species entering in. There's lots of ways that we know, there's lots of evidence that it's never complete, even though there's just as much evidence that, we found a lot, with there being about, an estimated 100 county records, these state records, and then finding these intellectual species. Like, I know that I collected a lot. I see these numbers, this is the amount of days I spent in the field, the amount of collections per square mile. But that being said, all of this coverage of the study area and this, great collecting effort is still not the whole picture. And, I put this picture here of these mountains, there's a plant that I could just accidentally walk over, which would be a whole new taxon introduction to the area. It's not likely, but it still exists and is probable. And so to kind of wrap up the project itself, the project results in the checklist, and with that checklist is a natural history section. And one of the great parts of working with the Kinnikinnick Native Plant Society was receiving one of their grants, which I was able to use to pay the illustrators that I was able to work with. So a part of wanting to work in the Selkirks was, there's a lot of interest in this area. There's a lot of recreators. And so I wanted to make a flora that people would use, not just land managers, but actual botanists. And I thought, if it's pretty, people might use it more. And so I know a few people, one of them, my last field tech, and was able to get the word out for a few scientific illustrators and was able to essentially consult with them through Zoom, actually, how to draw these botanically accurate illustrations. And so some of the funds from these grants that I received, and we'll talk about in a second, went to the artists' drawings. And I think we have a poll here, but these drawings are essentially going into the checklist themselves. And I'll talk about what the checklist looks like in a second. But this question here is, which of these species is the most common in the Selkirks, just to keep you on your toes. Yeah, and you also know, I didn't give you any preface for this, so it's really just a guess. I think this is good, but what I see, I'm gonna end this poll, and honestly, y'all are right. The most ubiquitous species, one of the most common species in the area is the Western red cedar, and I hope that you all can see why. Some of these drawings, they're absolutely gorgeous. Some are more accurate than others, but they really display a little bit of the beauty that I think a list doesn't quite capture. And so just to give you a little look into the list itself, these are some example entries. This is another one of the beautiful drawings from the wonderful artists. And included with each entry to the list will be the species name, the authority, the common name, which counties it's found in within the study area. A star indicates it wasn't collected in that county before, the abundance, and then what vegetation types it's found in, the elevational range, and then a reference specimen, which are all housed at the Idaho, the Stillinger Herbarium. And then this number refers to how many times I collected it. And then some of these species will have notes. Most of you probably know Botrychium viridi as Botrychium lanceolatum. And then there's other types of entries. You know, these characters are referring to it being noxious and introduced. This one's referring to it being a state listed species. And so we have some other examples of what these entries will look like. But to summarize, the implications why this project is important, you know, when these baseline inventories for geographic areas don't exist, it's really hard to understand what to save, what is important, especially when there are some, you know, different ideas around how to manage an area, but also when there are threats like development and climate change. So they're kind of invaluable, but it also fills our larger gap in just our knowledge of the Pacific Northwest. And so, you know, now we know a lot more about the Selkirk Mountains, but we also know a lot more about northern Idaho and then Idaho in general. And so seeing how that relates in a small scale and then the larger scale. But this list is really also just a huge gift to the conservation land managers of the region, the botanists and the recreators of that area. And beyond it, it's also an incredible data set. The majority of the species have a silica sample, which is leaf tissue that is preserved for DNA extraction. And those silica samples have already begun to be utilized by other researchers doing phylogeographic or biogeographic studies. And so there's kind of unending implications for the study. But one of the most important things I'd like to talk about is the fact that I was able to hire 3 femme and non-binary botanists to work with me over the last 2 years, and being able to have other botanists kind of there's a saying, you know, lift as we climb. You know, I didn't have a lot of mentorship before this opportunity. And so to be able to mentor other botanists in this work has been a pretty big deal. And I think that's just as important as the data. But I'd like to end with acknowledgements and then questions. I wouldn't be able to do this project without my advisors. I have a really wonderful committee, and then the field assistants themselves were pretty pivotal to this work. Some of you probably know some of this local guidance. Jennifer Costas Thompson with the the Panhandle National Forest Service. I also got a lot of really amazing support from in consultation through the tribes themselves to talk in order to create my natural history section. And then some other local historians, Tom White's at the Priest River Museum, Michael Lucid, and then Kyle Nagy with the Sandpoint Organic Agriculture Center. I also have some really amazing artists. And then, which I don't think anybody knows here, but one of them, Bo, was one of my field techs the first summer. And I also had an assemblage of funding throughout the years, which this project actually just wouldn't happen without. And that is my talk. I went a little bit over in time, and thank you all so much for coming. And without further ado, questions.
Hi, Harpo, I got a number of questions here. You're ready to answer. Here's a question that has 2 parts, semi-related. Was there any specimen you thought you would find but did not?
Yes.
Many.
Many. Oh, wow. I did not find Viola selkirkii. I think that's the one I beat myself up the most for. I was really excited about that plant because most of the collections of that plant they're either in fruit or the flowers wilting. And it's the only specimen with the species epithet Selkirkii, and it's in the Selkirks. And being a botany nerd, that was just something that I was really interested in. And I wasn't able to find it. There's only 2 historical collections for the region, and then the rest of the Viola selkirkii distribution is in Canada. And so that's one I think about a lot. There's also those sedges that Robert Bursick is just really amazing and was able to collect all of those really cool plants. But yeah, that's probably the top one. I'm pretty upset I didn't find. But I also wonder, is it even there, you know?
Right.
I was on hands and knees looking for a lot of these species and I didn't find them, you know? Sometimes it's timing, sometimes it's phenology, but sometimes they're not there.
The other part to that question was what was the biggest surprise, a specimen you were surprised to find that you didn't think you would come across?
I think I listed them, some of those montane species, you know, being like, wait, this is here, you know, because, you know, the Selkirks, before I went there, my understanding was like, you know, Western red cedar, coastal disjunct, temperate, you know, rainforest, you know, and so to have a lot more of these montane areas was pretty cool for me, actually. I mean, I got that Botrychium hesperium is pretty cool species. Finding that with moose this last summer was a pretty big highlight as well, because it was one of the plants. One of my mentors, Ben, you know, was basically like, these are the moonworts you might be able to find. And so I was very much in a place of like, all right, let's see if I can do it.
Another question was about referring to photo documentation in the field. Did you do that, and how did you do it? And
yeah, so the majority of those pictures are taken by me. And then I think there's maybe 1 or 2 taken by Moose over the summer. And then there's probably like 5 or 6 in the slides by Perry Lee Pipkin, who has a really nice camera and loves taking plant pictures. And that was also a really cool thing about the study, was just encouraging other people to take plant pictures, which was really awesome. But yeah, that's kind of a group effort there. But the majority of the pictures are from me, and they're really all from my iPhone. So yeah,
great.
that answers that question.
And you already talked about how amazed you were at finding 58 carex.
Oh, it's Bob Mosley. Yeah, Bob Mosley's collections are included in my checklist as well.
And he also asked about legumes. They're not in the top list.
Yeah, actually, Ben and I had talked about that. He was saying, you know, a more common family that would be more prevalent would be Fabaceae or Brassicaceae, but those families would be more common down south. So the fact that Rosaceae and Ericaceae were that diverse up here just really points to the more northern nature of the study area.
Okay, we have one more question. In what way did the Kalispel tribe help you? Was it in identifying only food and medicine plants? Did you learn about plants that you didn't know about prior to working with the tribal biologists?
Yeah, that's a good question. That's from Jane Fritz. I read your article. They advocated for me reading your article and using it in my thesis, which I do. I was able to, there is a tribal representative here at the university that connected me with the 2 main tribes, the Cooney and the Kalispel, and they each had representatives that I communicated with. And the way that they consulted me was in talking about their land tending history as it related to their presence within the Selkirks and general land practices within the area that have been historical, even if they're not currently using them. And also their history with being forcibly removed from the land, and they also pointed me to resources. And then they also verified information that I found, and I was able to basically have Zoom and phone calls with them in order to make sure that the way that I'm sharing their story was accurate.
Okay, and then there's one last question here. Have you used Marion Fuller's herbarium specimens in your work?
Yes, I have. She's got a lot of specimens. Most of her specimens are from the southern extent of the range or the lowlands, but definitely we love her collections and have been utilizing them within the area. And I think I have 1 or 2 maybe going into the checklist, but if anything, they were they're pretty much an amazing resource to guide some of the collecting here.
Good. Well, that's all the questions we've got. And I think what I'm going to try to do now is unmute everybody and they can give you a round of applause. Thank you for this impressive presentation, Harpo.
I'm sorry I was not able to practice this as much as I would like, but thank you for understanding.
Thank you very much. It was really, really great to hear.
Yeah. Somebody did ask me if I had any bear encounters. And I know that that's a very common question, so I'll just humor everybody by answering it. I think I saw 2 or 3 black bear each summer, but I didn't see any grizzlies. I did see, I think I had 2 mountain goat interactions, a few moose, and that was mostly it in terms of animal interactions. The mountain goats are the coolest, and the bears. I don't have any pictures because you can't really get that close, but and no wolves.
Good. Okay, well, let's give Harpo a round of applause, and thank you all for joining us today.
Those are my friends.
Thank you.